Alright, so welcome to another video from Shom's Biology. In this video lecture, we are going to talk about gel electrophoresis. And in this series of lectures, we are going to talk about gel electrophoresis. And the first lecture, this is all about the Agarose gel electrophoresis. We'll first talk about the basic principle of gel electrophoresis. Then we'll talk about the Agarose gel electrophoresis that utilizes the Agarose gel to separate mixture of DNA fragments, okay? And we'll also talk about SDS page. We'll also talk about pulse field gel electrophoresis. We'll also talk about the capillary gel electrophoresis in this series of videos, okay? So continue to watch them. So the very first thing that I want to talk about is about the gel electrophoresis principle. Now, to understand gel electrophoresis principle, you need to know one thing very simple, that when we say electrophoresis, we are trying to separate a mixture of charged molecules from each other. So we are dealing with molecules which are charged, right? So molecule without charge cannot be separated with electrophoresis, because electrophoresis by its meaning is separating a mixture of or resolving molecules based on the charge, okay? Charge to mass ratio, okay? So two things play a very important role. I'll take a color here first and then I'll explain. Two things play a very important role here. One is the net charge, okay, and the second thing is mass, okay, mass of that molecule, and we simply call it as a charge to mass ratio. This ratio plays vital role in separating a mixture of molecules from each other, and that molecule can be DNA, that molecule can be RNA, that molecule can be proteins. Now when I talk about the charge of all these molecules, we know one thing, that the DNA, RNA, they have this like phosphodiester bond in their backbone. So they have a negative charge by default, okay? But what about proteins? We know amino acids, R groups have different charges. There are positive charge R groups, there are negative charge R groups. But in reality, when there is a protein, most of the proteins, they don't have that kind of charge because they are made with multiple amino acids together. Some of them are plus, some of them are minus charge. So all over net charge of 0 or something, or very little charge differences, right? So when we are separating DNA and RNA, we have a default negative charge which helps the DNA and RNA to migrate towards positive charge. While we are separating proteins, we need to add some charge to it, okay? And what we do, we add negative charge to it, okay? We'll talk about that later in details. Now, once you understand that in electrophoresis, we always want to discuss about charge to mass ratio, try to understand the principle of electrophoresis. The idea stated is that in a field of electric, so this is, let's say, this can be any, let's say, beaker where a liquid environment is there, or it can be a solid state environment, which is agarose gel, whether the situation is liquid or a solid phase. But ultimately, if we apply electric field to it, okay, how can we apply it? Let's say we have a minus here and a plus here, two electrodes, right? And this is very important because many people have a mistake, a wrong conception here. So this plus charged or positively charged electrode is known as anode. Many people think this cathode, but it is anode that is positively charged, and cathode is negatively charged. Now people confuse with cation and anion. Cation is positively charged ion, anion is negatively charged ion, but anode is positively charged and cathode is negatively charged. Keep this in your mind. Now in this case, we have this gel, let's say a solid environment, we're talking about agarose gel, and two electrodes are placed, and let's say there are this DNA, and we know DNA already has a negative charge. So if we apply an electric field, I can change the color from here. So what we see, I took the color red, now black color. So when we apply an electric field, due to this electric field application, our molecule which we want to separate, it has its own charge. So based on that electric field, our charged molecule will migrate based on the charge that it carries, okay? It will migrate. Now this migration again depends on two factors, the net charge of that molecule and the mass of that molecule. How it is related? In very simple words, if the mass is less, means smaller molecules, they will migrate further. The mass is bigger, they will migrate slower. Now what about the charge? If it has more charge, carry more charge, migrate further. So let me say charge and migration is directly proportional and mass with migration inversely proportional. 1 by migration, inversely proportional. That's how it's related. So now when they are considering the net movement, or what we can call it as an electric force, based on which our charged molecule is moving from one electrode to the other electrode. The electric force, or you can simply write it as F electric. This electric force will be equal to, again, what it will be equal to? It will be equal to the net charge that our molecule carries. Okay, Q, let's say the charge is Q, and then multiplied by the net electric field that is applied. That is, let's say in this case the electric field is E. Okay, the electric field that we applied. So the force, the electric force that is applied to our molecules to move from one electrode to the other electrode depends on the charge and the electric field, Q into E. Now this is not simple, you understand this, right? It's very simple. Charge applied, the molecule will move. But this is not that simple, because as the molecule will move from one electrode to the other electrode, there is a frictional force applied by the environment itself. Either it's a liquid environment or solid environment. There is a frictional force. So we call it frictional force, or F frictional. Okay. So the frictional force is dependent on V into F. Now what do you mean by V and F? So this V is known as the rate of migration of the molecule, rate of migration of the charged molecule, and F is the frictional coefficient. Okay? So we know the frictional or electric force applied and force, the frictional force, both of them are repelling each other out because they are working in the opposite direction, right? So if we simply make this equal, then what we'll find out? So we can say, we can write it F electric equals to F frictional, right? Electric force equals to frictional force. So you can simply write it as QE, which is F electric's value, equals to frictional value, that is VF. Alright, so now we can rearrange it. Q by F equals to V by E. So this is something we are getting. Q by F equals to V by E. Got it? So now this is how we can derive the relationship of charge mass ratio. So in very simple words, what I can say from this equation as well, if our target molecule has less mass, it will migrate further. If it has more charge, it will migrate further. Okay. So if I compare a large molecule and a small molecule in a mixture with the help of electrophoresis, small one will move further, large one will move slowly. But on the other hand, if you're talking about the small one, let's say with very less charge, and the large one with very high negative charge, then as it has more negative charge, it will move further towards the plus end of the electrode. So not only the mass that matters in case of electrophoresis, the mass, charge to mass ratio, that matters a lot. Okay, so this is the idea of, this is the basic principle of any kind of electrophoresis. And there are two types of electrophoresis. Okay, one is the moving boundary electrophoresis. The other one is known as zone electrophoresis. So let me also change this slide and write this down. So the electrophoresis, I can write it here. The electrophoresis can be divided into these two different types: the moving boundary and zone electrophoresis. The moving boundary electrophoresis works in an environment which is liquid. Okay, so liquid environment, let's say a beaker or any tube where we put the electrodes, let's say minus and a plus, both electrodes, and this is liquid environment. So then there are particles in this liquid, charged particles. Based on the charge, they will migrate towards minus end or plus end, okay, as we apply the electric field. Now as this charged particles migrate towards plus or minus end, they continuously create a boundary of plus and minus in that environment, in that liquid environment or liquid media. And as this boundaries continues to change, as we apply the electric field, it continues to change depending upon the charge of the molecule that is present in the environment. That's why we call it moving boundary electrophoresis. While we have something as a solid environment, let's say a solid gel, and a solid gel means the gel has pores in it, but ultimately it's built with a solid material, that is agarose or polyacrylamide. So that gel can resolve mixture of DNA or proteins. So when we use for a solid medium, there is no change in boundaries because there is a fixed application. There is no change in boundary due to the solid nature of the medium. We call it as a zone electrophoresis. Okay, so that's the basic idea of electrophoresis. Now once you clear about the basic idea of electrophoresis, it's time for us to discuss about the agarose gel electrophoresis. So as I told you earlier that we'll be discussing about the agarose gel electrophoresis. So agarose gel electrophoresis is a type of zone gel electrophoresis method where it utilizes agarose to create the solid matrix. Okay, and the agarose is created means what? Agarose is basically is derived from seaweed. Okay, and the agarose is basically a polysaccharide of agarose. Okay, so polysaccharide. It's a polysaccharide. That means basically sugar molecule. So when we take this agarose and we allow it to form, like solidify, then it creates what is known as a network like structure, a mesh like structure like this. And this mesh has bigger pores in it. It creates mesh, but the pore size is big. So agarose's pore size, the pore size is big. So any molecule, even more than 100 kilodalton, 200 kilodalton of mass can be separated with agarose gel electrophoresis. Bigger molecules can be separated because the pore size is big. They can easily enter the pore and exit the pore, right? Quite logical. There is nothing to be extraordinary here. Okay. Now, one more thing that you want to understand here is that in this case, this agarose can hold on to like 50 kilobase sequence, up to 50 kilobases length of the DNA to be separated. So if you want to separate larger DNA, we can separate until 50 kilobase long DNA sequence using agarose gel electrophoresis. We can still extend it to 10 MB length of the DNA with little bit modification of the same agarose utilization, but little bit modification of electrode placement. We call it pulse field gel electrophoresis, which we'll discuss later on. So what happened with agarose is that generally we use 1 to 3% of agarose to create this gel, solidifies it, and then we load the DNA. We create grooves and we load the DNA. Let's say this is what is known as a marker. Marker means basically known length of fragment of the DNA is read here as a marker, like a scale. Okay, and then we load our target DNA mixture, and then the mixture gets separated, T1, T2, T3, let's say here, like this. So what we can say, based on this, let's say this is negative charge, this is positive charge. Okay, and in agarose gel electrophoresis we are separating DNA, and as I told you, DNA due to its phosphate backbone has a negative charge, right? So as DNA has a negative charge, DNA will be separated based on the movement of DNA towards the positive charge of electrode, and this is the positive charge of the electrode. So DNA will move from negative charge to the positive charge. And as the DNA will move, what we can say is that the smaller length of the DNA will migrate further. So this is smallest here as per this picture, right? And the biggest as per this picture, let's say here, this is the biggest, okay? So bigger length of the DNA will migrate slow. That is the idea, because the net charge we consider the same due to the phosphate backbone. Net charge is the same, they are separated based on the length. So Agarose gel electrophoresis of the DNA actually separates DNA based on its size, length, or we can say the mass, obviously, because length is directly proportional to the mass of the DNA, right? So they are separating based on the mass, or you can say based on the length of the DNA. It can be separated quite easily, okay? And now once we separate them, the separation is done. After the separation is done, what we need to do? We need to visualize it, because we load it, we separate the gel. We cannot visualize the DNA, right? We need to add some dye or something to visualize it. So utilize different kinds of dyes. So I'll change the color here as well. A red color again. We use different dyes, different type of dyes to visualize the presence of the DNA in the agarose gel. What we add? We add acridine orange, okay? We add ethidium bromide or acridine orange, okay? These are the common dyes that we use. Generally, they are the base intercalating agents. So if we consider DNA as a ladder shaped structure like this, so these dyes, they stack themselves between the base pairs of the DNA. And once we add them, for example, ethidium bromide, ETBr, once we add it, and then we put this whole agarose gel plate in a UV chamber, then this DNA can be visualized as an orange color due to the presence of the UV light. So we can easily view the bands in the DNA with orange color. We can take a snapshot of it, and then we can understand the length of the DNA in our mixture. That's how we run agarose gel electrophoresis. Okay, and remember, this gel electrophoresis, this also again depend on the percentage of agarose that is used, depending upon whether we want bigger length of the DNA to be separated or smaller length of the DNA to be separated as a whole. Okay, and if we don't know anything, then we generally keep it from 1% to the 3% range, not more than that. Okay, so that is the idea of agarose gel electrophoresis for DNA. One last thing that I want to discuss before closing this idea of agarose gel electrophoresis of the DNA is not only, as I mentioned, the DNA is separated based on the mass, means the length of the DNA. But apart from the length, the compactness of the DNA matters a lot, right? Why? Because we know DNA can be present in different secondary structures, okay? And the secondary structures of the DNA means like the DNA forms supercoils, right? Simply, if we talk about the circular DNA, closed circular DNA, we call it CC DNA, closed circular DNA. Or we can say the covalently closed circular DNA. We write it covalently closed circular DNA, CCC DNA. Now this is very compact size of the DNA, but this is just like a plasmid like structure, covalently closed circular DNA like this. Now this DNA may have a supercoiling like this, or it may have a supercoiling like this, multiple supercoiling like this. So the more supercoiling the covalently closed circular DNA has, the more compact it becomes. More compact means it can migrate through the gel faster. This is something different. The same length of the DNA length remains the same, but it has multiple rides. Now what is ride, what is twist angle and everything, if you want to know, you need to watch my lecture on the supercoiling of the DNA. But now we are talking about it. So the more complex the DNA here is, the more supercoiling is. They're like topoisomers, like they are known as topoisomers, because they have the same length, but they have different like secondary structures to the DNA. So they are known as topoisomers of each other. So if you are separating topoisomers of DNA from each other, then more compact the DNA has a higher and faster resolution in the agarose gel. This is something that you need to keep in your mind. The open single-stranded DNA will find it difficult to migrate through. Closed circular DNA will migrate faster than linear DNA. And even between closed circular DNA, if it has secondary structures, then it will migrate further. Okay, this is something that you put in your mind. That's almost everything that you need to know to answer most of the questions from this topic, that is gel electrophoresis of DNA. Agarose gel electrophoresis of DNA. If you like this video, please hit the like button, share this video with your friends, and subscribe to this channel to get more videos like that in future. Thank you. Bye.